High school biology textbooks follow a predictable script. A streamlined sperm meets a massive egg, they fuse, and a new life begins. But this simple picture masks a series of fierce scientific controversies. The gametes driving this process are actually the subjects of three massive, unresolved debates. To map the future of human reproduction, we have to look at how these sex cells evolved, why they mechanically fail, and whether we should start manufacturing them in the lab. The foundations of reproduction represent an active, fiercely contested frontier where rules are currently being rewritten. Biologically, the sexes are defined by a single metric called anisogamy, the extreme size and mobility difference between gametes. By volume, the human egg dwarfs the sperm cell by a ratio of roughly 2,000 to 1. This disparity presents a paradox. Our ancient ancestors reproduced using isogamy, meaning their fusing gametes were identical in size. Evolution eventually forced life into two distinct extremes. Parker's parasite-host model treats gamete production as a strict economic trade-off. A parent has a fixed energy budget. They can either produce a few massive, nutrient-rich cells or shatter that budget into hundreds of tiny, fast-moving ones. Over millions of years, the tiny gametes evolved to numerically overwhelm the large ones. One sex became an evolutionary parasite, specializing in locating as many targets as possible, while the other sex became the host, specializing in provisioning the zygote with nutrients. Alternatively, the organelle conflict hypothesis suggests the sperm shrank to solve a cellular war. If both gametes contributed cytoplasm, their competing mitochondria would battle for dominance. Sperm discarded their cytoplasm to ensure a peaceful uniparental inheritance of mitochondria. Nature occasionally breaks these rules. Male honeybees develop from unfertilized eggs, while the female-only whiptail lizard reproduces asexually, while still mimicking ancestral mating behaviors to trigger ovulation. Distinct sexes and gametes persist today as the surviving outcomes of this ancient, microscopic arms race. While that arms race is ancient, its mechanical failures are a present-day crisis. Today, a massive percentage of male infertility is classified as idiopathic, or completely unexplained. Much of this failure traces back to oxidative stress. Unstable reactive molecules silently accumulate within the reproductive tract, degrading internal membranes and fracturing its DNA. This has sparked a debate over clinical standards. While many doctors prescribe antioxidant supplements empirically, researchers argue for precise biomarker profiling to identify the specific oxidative phenotype before treatment begins. These mechanical thresholds are debated across all kingdoms of life. For decades, botanists could not agree on how plant gametes manage to synchronize their internal cycles for a successful fusion. In these panels, the glowing punctate foci reveal the exact state of mature Arabidopsis pollen. They show the male gametes are held in a pre-replication arrest until the moment they are delivered to the ovule. Pan across this multi-panel microscopy of isolated egg and central cells. The exact same pre-replication arrest is present here, ensuring the female gametes remain in a state of suspended animation until the sperm arrives. From human clinics to botanical laboratories, fertilization is revealed as a fragile mechanical threshold, far more complex than a simple cellular meeting. If mechanical science shows us how fertilization fails, a new technological frontier is asking what happens when we manufacture the process from scratch. The technology is in vitro gametogenesis, or IVG. It allows scientists to take a raw stem cell and artificially force it to transform, potentially turning a skin cell into a functional human gamete. IVG could provide biological children for individuals who lack ovaries or testes, and it would enable same-sex couples to have offspring genetically related to both partners. But the risks are unprecedented. We have no data on the epigenetic abnormalities IVG might trigger. It risks the commodification of superior cell lines and opens the door to solo reproduction, creating a child from a single person's DNA. The most critical debate involves consent. IVG introduces the possibility of unauthorized reproduction, using stolen genetic material to create a child without an individual's knowledge, even after their death. This technology detaches reproduction from the human body, transforming biological legacy into a manufacturable technology. The study of gametes and fertilization is currently rewriting the most basic rules of life. By mastering these mechanics, answering why they evolved and how they fail, We are entering an era of human capability that sidesteps millions of years of biological constraints. The core conflict shifts from the mechanics of how life is made to the authority of who gets to make it, and the boundaries we must establish before crossing the point of no return.